So welcome. I am Hashir, and today I'll be presenting my project titled Protein Function Detection Using Machine Learning. So this project explores how machine learning techniques can be used to predict the molecular functions of proteins based solely on their amino acid sequences without the need for any complex 3D structural data. Proteins are fundamental to all the biological systems, performing diverse roles as catalysis and structural support. Understanding their function is critical in areas like drug discovery and bioinformatics. So, this project focuses on predicting the protein molecular functions using features like which are derived from their primary sequences. So by extracting the sequence-based features such as amino acid composition, sequence length, and dipeptide frequencies, and applying ML models like random forest and XGBoost, it will be used these two models, uh, both fine-tuned using grid search CV. For XGBoost, we did that to improve slight accuracy, and we aim to provide accurate and efficient prediction without relying on 3D structural data. So these are the contents presented in my PPT. So we'll start with an introduction to the topic and then look at the project's objectives. And then I'll follow that with some background on recent approaches and then discuss the methods I used, including the data, features, and models. Then I will present the current progress and end with a summary and key references. So proteins are central to all the cellular activities like metabolism, signaling, and transport. So predicting their function helps us understand diseases better and discover new drugs. And rather than relying on 3D structures, which can be hard and expensive, uh, we are using readily available primary sequences. We extract useful features from these sequences and applying the machine learning models to classify their molecular functions. And this approach is not only efficient but also it is scalable across large data sets. So comes to the objectives of this project. So the goals of this project are first of all to build a machine learning model that can predict 14 functions. Second is to clarify which structural or sequence derived features contribute most to the accurate predictions. And next, third one, to compare the performance of different HTML algorithms. And finally, to evaluate the effectiveness of these models when applied to sequence-only data without needing the structural information. In recent years, tools like Boost have significantly advanced protein function prediction. And come to Boost, it combines PyBoost. PyBoost is basically this like a Python-based implementation for gradient boosting. Simply an ML technique which is useful for classification, regression, and ranking the tasks, etc. So it combines the proposed combines both the boost with the GNN graph neural networks. That it is a deep learning technique. Uh, this proposed so this model achieved second place in CAFA5. This is a coding challenge competition based on protein function prediction. So this means it shows the power of deep learning, especially when combined with the structural and sequence information. Uh, so also it shows that the models like CNNs, transformers, and GNN are becoming increasingly prominent in functional annotation, discussing the importance of multimodal learning strategies in bioinformatics. So, so another model uh, is worth noting, which is Panda 2. It integrates the graph neural networks with the transformer-based reporting language models. So this Panda 2 stands out because it can predict across all the three categories of geo, which is gene ontology, which are biological process, cellular components, and molecular functions. So by capturing both the sequence patterns and structural relationships, it achieves a greater performance, demonstrating the benefits of combining the diverse deep learning strategies for comprehensive protein annotation. So for datasets, I took dataset from coding challenge, which is CFA3. So CFA3 training dataset was available in the website, and also we used a dataset from Uname for database, but we haven't implemented it yet. So, but these datasets provide high quality protein sequences in FASTA format, along with corresponding functional annotation in a text file, which was GU in a text file, and our focus was on proteins with experimental validation, so as to ensure the accurate training and testing of that hormone. So, in terms of feature extraction, I used several key sequence-based features, which includes amino acid composition, the total sequence length, and dipeptide frequencies, which is still in progress. We haven't completed it yet. Uh, and these features help the model learn patterns in the sequences that are associated with the specific functions. So, for the modeling part, I took two machine learning models, which are XGBoost and RNForest. So we used these two models till now, and these models are known for their robustness and performance on structured data. So to evaluate their performance, I used standard metrics like accuracy, precision, recall, and difference score, which would give a balanced view of how well the models are doing across different protein functions. I did this project entirely in Python using standard scientific libraries or tools like scikit-learn for ML algorithms, pandas for data handling, and biopython for working with the biological data formats. Also, biopython in particular was useful for parsing and manipulating the FASTA file we use database, and also these tools provide a solid and flexible foundation for building and testing these machine learning workflows in bioinformatics. So the data set used is a subset of unique process for database provided as a part of the CFA3 training data that we have discussed before, and it contains experimentally validated protein sequences in faster dot. And each protein is mapped to the GO terms, which is gene ontology terms, specifically from the molecular function F category. And for this project, we focused on the top 10 most frequent GO terms, which resulted in approximately 9,500 protein samples. And the extracted features includes amino acid frequencies, sequence length, and dipeptide composition for what's also used in machine learning models. So far, I've trained the two models. They are random forest classifier. I used 30 estimators because of some certain memory issues. and a maximum depth of 10 value, and it achieved a accuracy score around 24%. It was pretty modest. And uh, the next model that I've used was XGBoost Classifier using 100 estimators and learning rate point 1, max step value 6, and it performed slightly better, like only 25%. Its accuracy score was around 25%, and but it showed like improved F1 scores for specific geoterms that is mentioned in the slide. You can see, and that it means it indicates that it's learning meaningful patterns even if the overall accuracy seems modest. So also we are currently working on a deep learning model so as to increase its accuracy score. These are the necessary inputs needed for this model. We use pandas, Python, and this is our model. We used to train. We used the ML model and classification report which contains all the metrics we needed to evaluate our model. So we start by reading protein sequences in FASTA format using Python, extracting both entry ID and the sequence itself. And then we load a separate annotation file that maps each protein to a GOT, filtered specifically for molecular function category. Next, we merge both the data sets on entry ID, and then for feature extraction, we calculate the frequency of each of the 20 standard amino acids and the sequence length for every protein. This gives a simple and informative feature set. Then we filter. Then we filter the dataset to include only the top 10 most frequent geotags to ensure the class norms and to manage this our model's complexity. Then our model are encoded. I mean, like the, well, the model registers are encoded into numerical labels using labeling. Then we perform a standard train test split with a ratio of 80 by 20, which means 80 for training and 20 for test. And the random forest plus random forest is configured with 30 estimators. Default was 100, the default earlier was 100, but when I used 100, it was constantly crashing. So I reduced the estimator value. And max step value is I give 10 to 74 value. And after training, we evaluate the model using metrics such as the precision, recall, and effort score. And then this we say like a feature importance plot showing the top and most influential amino acid and sequence length. Okay, this. Now already ran this project. So okay, this is the top and most impressive features, and this is the accuracy we obtained. So this is a random forest, and next we have XGBoost. So when also in XGBoost we used pandas by Python and the similar model XGBoost and similar classification report and label encode of. So As with Granapol's model, we started by loading the protein sequences from a FASTA file and mapping them to geomolecular function terms from a tab separated annotation flow. After merging these on after merging this on entry ID, so after merging on entry ID, we processed it to an enhanced feature extraction process. So here, once all the features are extracted, we filter the data. We fi- before that, in addition to like in XCBoost, we have also used uh, dipeptide frequencies in addition to the amino acid frequencies and sequence length in XGBoost, which is a combination of two amino acids, which greatly increases the feature richness and allows the model to learn more sequence patterns. So once all the features are extracted, we filter the data down to top 10 most frequent geoterms and encode them using the our label encoder. So the yeah, so and we use the label encoder to encode them use like and we split the data set into training and test data in an 80/20 ratio. Likewise, we did in random forest. So as to optimize the performance, we performed a search using grid search CV. This is simple. We used grid search CV over several hyperparameters like max learning rate and number of estimators, which are the parameters used in XGBoost. Using a stratified cross validation to prevent over 50 and serve the class distribution. So once the test configuration is on, we train our model. We train our model. Mm, this is our model is trained. And after training our model is as usual evaluated using the metrics, which is including classification, include which includes the accuracy, precision, recall, etc. And also we also visually top 10 most important features. Uh, so hence in this project actually develop ML models that predicts the protein functions using only sequence-based features. So by using this amino acid composition and sequence length, we achieved some good results without relying on structural data. So this approach could significantly benefit from the bioinformatics, especially in areas like drug discovery or drug development and enzyme engineering, where this a rapid large-scale function prediction is essential. So these are the key references I have used throughout my entire project. The datasets are used from CAFA and UniPro, while the methodologies and background were used from recent studies like DeepGo and the work by Avery on biomolecules. So all these resources were useful and it provided a solid foundation for building and validating my models. So that's all about this project. Thank you.